So next I will introduce Professor Tania Sorrell. And Tanya will speak about lessons from infectious diseases. Tania Sorrell is a professor of clinical infectious diseases, director of the Marie Bashir Institute for Infectious Diseases and Biosecurity, and a deputy dean of the Sydney Medical School of the University of Sydney. She has had a long-standing clinical interest in mycology and infections in the immunocompromised host, and a more recent interest in emerging infectious diseases. Her research has focused on the pathogenesis of fungal infections, emerging fungal diseases, and new antifungal drug development, new diagnostics and clinical trials of antifungal diagnostic and treatment strategies. She has served on state and national advisory committees in infectious diseases and therapeutics and the research and human ethics committees of NHMRC. Thank you, Tanya, for joining us today. I'll give the word to you.
Thank you very much for a very kind introduction, and good morning, everybody. It's a pleasure to be able to speak about the infancy of precision medicine in infectious diseases. And I'd like to just outline briefly what I'll talk to you about, which is the actual context and scope, which does differ from chronic diseases, to look at where we are in 2020 and where we might be in the future. So there's an extra dimension, of course, in infectious diseases, and that's the advent of a pathogen that interacts with a host. But not only that, there is an environment which can be external, but also internal in terms of the microbiome. And there's a human population where infectious diseases can actually spread from person to person. And so what I'm actually going to do today is distinguish between what we've perhaps over enthusiastically called precision public health, but also personalized health care, which is no doubt what you were expecting me to talk about. In the context of genomics, which has really revolutionized opportunities for infectious disease and microbiology, and that's the genome of SARS-CoV-2 up on the right of the diagram, there are a number of areas where genomics has made an enormous difference. It's helped with pathogen discovery, and of course, COVID is a very good example of that, looking at evolution, looking at the origin and actual spread of, in this case, a virus, and defining diagnostics based on the genome that can help us track infection, but also develop serological tests to do tracking. On the other hand, in terms of an individual patient, we can not only develop microbial diagnostics, but look at some of the genomic factors that have already been referred to in terms of other diseases. And not only that, think about whether we can actually do prognostics, that is to say, use genomic responses to infectious diseases to tell what the outcome for a patient's likely to be and how we might triage them in the clinic. And you've already heard about pharmacogenomics, which is clearly a burgeoning area, or about to be, in infectious diseases as well as in other contexts. I'd like to thank Eddie Holmes for supplying this diagram of the evolution of SARS-CoV-2, and it points out some very interesting information. It was probably back in November of 2019 that the likely common ancestor arose, and fairly quickly two main lineages actually developed, and I don't seem to be able to use the mouse, so I'll have to use descriptors. If you look at the A and B, these were the two lineages that separated quite early in Wuhan, but we've now seen more than 100 lineages globally. B1, which is the largest part of the light blue in the middle column on the right, arose in Italy. And in Australia, our two waves have been associated actually with B4, and with what was originally part of B1, but is now called D2. And that second wave is, of course, the outbreak that we've been describing in Victoria. It's important to be able to look at these evolutionary trends for many reasons, but in the context of this talk, one of them relates to public health and prevention of spread and tracking of spread of the organism. There's another one that relates to diagnostics, because the molecular diagnostics that we actually use in this condition have really taken out just a few of the genes specific to this virus. And if a mutation arises in one or more of those, then our diagnostics are really no longer, are completely fallible and may in fact be not helpful. So very important for us to do this monitoring. But I'd like to give you a practical example, and all of the examples I'm using today are actually from Australian research, with the exception of one. This was a very nice study that was done in Vitaly Sinchenko's lab in Sydney, and it was done during the first cases that were seen in New South Wales between January and March of this year. The concept was to employ genomics to look at tracing the outbreak and whether or not clusters could be identified over and above those which regular contact tracing could accomplish. So if you look at the diagram on the left, the inner circle, the different colors of the bars in the inner circle actually represent the different global lineages of the virus that were present at the time. The outer circle represents the origin of the infection of the cases in New South Wales, with the pale blue, which is making up most of the color of that circle, representing overseas acquired infection, the bright yellow representing locally transmitted infection, but with an epidemiological link on the basis of contact tracing. And if you look within the middle of that diagram at the tree trunk and the clusters that are evident there in green, and I would perhaps point out that the pink cluster is the Ruby Princess cluster, but within the green clusters, there are solid green dots, but there are a few yellow dots that you may be able to see that I've highlighted by arrows. And they represent genomic data which built on the regular epidemiological data and in fact identified an additional 40 clusters. So it was who not previously had a known contact. So genomics enhanced contact tracing is a very practical outcome. These additional cases were identified both in travellers but also enabled identification of likely sources. And the validity of this approach was actually supported by computer simulation and agent-based modelling. So perhaps I'll now move on to what you probably did expect me to talk about. And we've heard about this right drug, right place, right time from one of the previous speakers. In infectious diseases, there is perhaps one difference from most of the chronic diseases, and that is urgency. With severe infections like sepsis, we haven't got time to do genomics and do analyses and come back with an answer a few days later. Sepsis itself, associated with infection, accounts for up to 50% of hospital deaths. Mortality in severe sepsis rises at about 8% per hour. And in the presence of AMR, mortality from sepsis is increased by about 5 to 12 times, AMR, of course, being antimicrobial resistance. So in a severely ill patient, we traditionally combine a number of clinical factors, as I've illustrated in the diagram there, together with local microbiological epidemiological knowledge about antimicrobial resistance. And we institute best guess empiric therapy, which may or may not work. Most of the time it does seem to have an impact, but it also creates problems in terms of if it's used very frequently, the likelihood of further emergence of resistance or adverse drug reactions in an individual patient. But nevertheless, it's obviously important while we're awaiting a microbiological diagnosis. Now, from the left of this diagram, you look at traditional diagnosis. Of course, we normally like to have a pure culture of an organism. We may try to identify or speciate it further using biochemical methods, or we may put blood directly into blood culture bottles. But any of these have created a time lag in terms of either amplifying the organism so we can identify it more quickly, or actually being able to identify it microscopically. And then in order to actually understand the antimicrobial resistance pattern, it's another several hours or 24 hours in traditional microbiology. On the other hand, with the advent of PCR-based techniques, we can now take a blood culture, amplify it maybe for 6 hours or so with a rapidly growing organism, do a PCR within about 3 hours, and where you can see that we're increasing the rapidity of the diagnosis. But in 2020, in fact, genomics can inform a lot more than that. And this happens to be work that is done by John Idel's group, again in Australia, where they've used what's called a multiplex PCR approach, where you can actually identify up to or more than 70 different targets in the one assay. PCR, of course, will pick up what you're looking for if it's there. So what he's tended to do is think about the most common organisms that are likely to occur in a blood culture and cause sepsis and make sure all the targets for those are there. But he's also done some interesting work which has been confirmed in animal models, looking at genomics to identify bacterial load using the 16S rDNA gene, which is the one in bacteria that we use. And within 3.5 hours of actually collecting blood, he can come up with a bacterial load. Now that's important because the load of the bacteria correlates with mortality or outcome. But not only that, in gram-negative bacteria and gram-positive bacteria, the major resistance genes are actually constitutively expressed. So we can use genetic methods to identify those as well. In my area, in fungal infections, that's actually not always the case. We have to look at transcribed genes if we can identify associated genes at all. But John's group did some very interesting work on AMR where they actually found very quickly, within just a few hours, with a very high negative predictive value, they could look at Sydney sepsis patients and be able to much better target the type of therapy that they were planning to select. The other side of the coin, of course, is in the host and triaging the host so that we can give them appropriate treatment. And in the 20th century, there was a lot of interest in developing clinical risk prediction models, which are linked with imaging and other tests, and we've heard a little bit about the imaging side earlier today. But with genomics, we can now think about a number of other things. Now, I was interested in your comment, Chris, about the pre-genomic testing, if you like. In other words, understanding inherited diseases that might put a patient at risk of infections. What I want to focus on is actually the prognostic implications. Can we use genomics for outcome prediction and not talk about pharmacogenomics because it's really not utilized yet, but should be and could be in infectious diseases in terms of drug metabolizing enzymes and adverse drug reactions, as has already been mentioned in other contexts. There's a very interesting example here, again from Australian-based people working with overseas collaborators, trying to define blood biomarkers that might be predictive of prognosis and triage of patients with respiratory infection caused by influenza. And in other words, can we use these to inform the need for escalation of care? And you can see that in the plot on the left, in fact, 128 upregulated genes were identified in patients with more severe influenza. A multi-way importance plot was then done trying to dissect out those that were actually making more of a difference than others. And in blue, 15 genes were actually identified. They developed a gene score in relation to this, which really just simply looked at the difference between the upregulated genes and the downregulation genes. And they did an international study in many countries, or employing patients in many countries, presenting to emergency department with influenza. Their risk prediction model was a clinical one, which I alluded to previously, versus their genomic model versus both. And the endpoint was the need for oxygen therapy at 24 hours. And you can see down in the right-hand corner, the clinical score gave a sensitivity of 83% and a specificity that was very low. But when combined with the gene score, the sensitivity was much improved, as was the specificity. So I think this is a really interesting application that we can use both microbial and host genomic data to actually fit into our clinical models. And there are many data streams, of course, that we need then to connect to do that. The patient's clinical signs themselves, perhaps some background risk factors, the genomics assays, the host signature assays, combined in the data integration and reporting platform, which can very quickly guide clinicians with where to triage the patient to and which drug to actually treat them with. And my final slide is really to do with, well, that all sounds fine, but is there a future where we actually use directly on biological specimens metagenomic or metatranscriptomic information? And again, of course, a biological sample in infectious diseases has all of the genes associated with pathogens that might be there, or in samples that are not sterile normally, like respiratory samples, there will be a microbiome there. And there will also be host genes, which are either expressed, transcribed, or not transcribed in relation to the presence of microorganisms. So what I'm inferring here, of course, is there a way we can take all that data, analyze it comprehensively, and come up with a pathogen and an antimicrobial resistance pattern, whether or not there is a co-infection present, whether or not the issue is a colonizing microorganism versus an infective microorganism, and whether we can actually, using our host genomic data, actually decide that this isn't an infection at all, it's a septic patient or a very sick patient for another reason. And so the diagram on the right is a representation from a really interesting paper by Langelier and his colleagues, published in 2019. And what they were trying to illustrate here in a series of patients in intensive care units who were having tracheal aspirates was to use both metagenomics and metatranscriptomics to see if they could not only improve the clinician-directed diagnostic performance of their assays with the use of these metagenomic techniques, but whether or not they could also distinguish between an infection causing the patient's critical illness versus not infection, and whether or not there was something in the microbiome that could actually contribute as a third partner, if you like, in this ultimate analysis. What the diagram on the right shows, in fact, is in relation to the microbial pathogens, and they did a lot of analytics in terms of, if you look at the top histogram, that's Streptococcus pneumoniae. We know that's a common cause of pneumonia. In the bright red section of that histogram is the clinician-driven diagnostic determination that Strep pneumoniae is present in that patient. The dark brown extension of the histogram is actually the additional identification of Strep pneumoniae that was made by using these genomic techniques. Now, Strep pneumoniae is an interesting one to talk about because you might think, well, you've got a patient in ICU, you found a Strep pneumoniae, it's got to be the pathogen. One of the issues with both Strep pneumoniae and Haemophilus influenzae is that they colonize the nasopharynx or the respiratory system without necessarily causing disease. And in fact, there may be co-pathogens there. So an additional dimension is to actually then try and look at whether or not this is a true pathogen using a couple of methods. One is the metagenome, because if there is actually a true infection, the diversity, not the metagenome, sorry, the microbiome, the diversity in the background microbiome is reduced. And these workers were actually very, I thought, intriguingly able to combine that reduced microbiome score, if you like, with the pathogen to infer that it was more likely to be the cause of the problem in the ICU. But not only that, they also, from the same sample, were able to look at some of those host genetic markers that I was talking about. And they actually viewed this as a triage process in itself, because if there were genomic markers that could be attributed to a viral or a bacterial infection, and there are ones that specify that, as I indicated in Ben Tang's data, then that would suggest that it is truly an infectious process. But there's also a set of sepsis markers, if you like, that are associated with non-infective causes of sepsis. So combining all of these three, which seems to be a mind-boggling proposition as far as I'm concerned, they were actually able very accurately to define the pathogen cause of the problem, and if it was not pathogen caused, then of course antimicrobial agents could be ceased. This concept is clearly, I think, exciting. If it's possible, is another matter. I should say that at the moment we can only diagnose about 40% of cases of pneumonia with the pathogenic cause, and you can see that already using these kinds of microbial information, that could actually be increased up to 50 to 60%. But as an overall construct, looking at all elements by unbiased sequencing, I think we've got 5 or 10 years to wait. So with that, I'd like to thank you for your attention, and if there's time, take any questions.